All right, good morning, everyone. Thank you, Al, for that really nice introduction. My name is Gina Yosten. I am a professor of pharmacology and physiology at St. Louis University, and it is my pleasure to chair this first session on the genetic and gene expression studies that aim to understand the cause of glucagon secretion defects in diabetes. Our first speaker for today is Lori Sussel. Lori is a professor of pediatrics and cell and developmental biology, the Sussel and Finlow Stem Cell Chair, and the director of the Basic and Translational Research at the Barbara Davis Center for Diabetes in University of Colorado. So with that, let's welcome Lori.
Thank you. And I want to thank the organizers for inviting me and also giving me the opportunity to talk about, there's me rather than my slides, actually. Are we going to be able to show the slides? Okay, that's much better. That was sort of scary, actually. So yeah, I have the opportunity to kick this off, and I call this alpha-beta soup because we're going to talk about transcription factors, and a lot of people tell me that when I talk about those, it's alphabet soup, but today it's alpha and beta soup. So I'm going to just kick off with little bit of an introduction since I'm the first speaker, but obviously this is an audience that doesn't need, much introduction to the pancreas, but as we all know, it's a multifunctional organ, and obviously the focus has been mostly on the beta cells that make insulin, but what we're here today is talk about the alpha cells that make glucagon. We've learned a lot over the years about the endocrine functions, and as Al said, we know a lot about the beta cell and how it's involved in hyperglycemia and lowering blood glucose levels. But today we want to focus on the alpha cell. And again, as Al said, in conditions of hypoglycemia, we get glucagon secretion and we get the increased blood glucose levels. And like I said, the focus until recently has really been on how the beta cell becomes dysfunctional in diabetes. But really what we want to focus on is the alpha cell today. And there was this really nice study actually by Al and Marcella, who they showed that in type 1 diabetes, the alpha cell actually, either in cases of extreme beta cell loss or when you have systemic hyperglycemia, you get dysregulation of the alpha cell. And what I think they really nicely showed in this, looking at human patients, is that there was altered alpha cell gene expression, and this led to downstream impaired glucose, or glucagon secretion. And they showed really nicely that there was a number of different transcription factors that were actually downregulated in the islets of type 1 diabetic patients, including MAFB, which is one we know a master regulator of alpha cell identity and function, and a downregulation of ARCs, another master regulator. But interestingly, there was also an upregulation of NKX6.1, which is actually a beta cell gene. So these cells were not only losing their function, but it seemed like they were losing their identity. And they went on to show that a number of different genes were downregulated. Alpha cell genes were downregulated, and beta cell and stress genes were upregulated. But I just want to focus on, since today I'm going to talk about transcriptional regulation. They also showed, again, a number of these alpha cell transcription factors were downregulated, and then we had this upregulation of NKX6.1, a beta cell gene. So this is, to us, really intriguing because I've been studying transcriptional regulation for a long time, but it was really a surprise that not only was there dysfunctional regulation of the beta cell, but also of alpha cell identity. So, like I said, transcription factors have been what we've studied for almost all my career. We've been trying to figure out how these transcription factors regulate islet cell fate decisions. And it's not only do they regulate them during development, it turns out that a lot of them need to continue to be maintained in adults, and if they're not maintained in their expression, they actually affect the function of the cell and the identity of the cell. And in particular, we were looking at NKX2.2, which was one of the transcription factors that Marcella saw was downregulated in the alpha cell. The interesting thing about NKX2.2 is it's a homeodomain transcription factor. It's expressed here, you can see in the embryo, it's expressed in its CNS, which is actually where I started studying it, but it's also expressed in the pancreas. And it's essential for both the development and the function of the cells where it's expressed. Interestingly, mutations in human NKX2.2 cause neonatal diabetes and neurological disorders. And these patients, these infants, have no detectable C-peptide, which is very similar to what I'll show you in the mouse model. But they can't actually measure, or they didn't actually measure glucagon. So we don't know what's happening to the alpha cells in these humans. So obviously we use mouse mostly as a model system so that we can figure out what's happening or how to make an alpha cell. And we know from studies that actually I did a long time ago that here's just a P0 pancreas, that when we knock out NKX2.2, it's pretty dramatic. We have absolutely no beta cells and very few alpha cells. And so what we know from a number of studies is that NKX2.2 is absolutely essential for promoting both the beta sulfate and the alpha sulfate. Whoops, wrong way. Okay. And so just to emphasize here, so this is really dramatic because we know that there are alpha cell and beta cell specific transcription factors that are only expressed in one lineage or another. So it's easy to understand how these transcription factors might be regulating those cell fates. But what about NKX2.2? It's expressed in both cell types. So how is it actually regulating two very closely related cell types and maintaining their regulation in the adult? So again, it's downregulated in people with diabetes, both in the alpha cell and the beta cell. So how is it actually functioning? And so this is sort of where I wanted to focus our studies on. Obviously, we could say there's the usual suspects, that there's different isoforms of the transcription factor. There's not. There's only 2 exons, so there's not a lot of isoform differences you can actually have. We have not been able to identify any post-translational modifications that are specific to a cell type. And we've actually done a lot of work looking for cofactors by doing mass spec analysis. Nothing has come out that actually tells us that this transcription factor is interacting with partners in one cell type versus the other. So, as I said, we've studied this a lot in beta cells, and we know both in the beta cell that NKX2.2 is important for the development, but it's also when we knock it out in the adult beta cell, it's required for both the maturation and the maintenance of beta cell identity. And we published this a number of years ago, but what we found was that when you knocked out NKX2.2 in the beta cell, we lost beta cell genes and we activated non-beta cell endocrine genes, so the glucagon pathway and the alpha cell pathway and the somatostatin pathway. And what Giselle, the student in the lab, showed was this was because NKX2.2 is largely interacting with enhancer regions to regulate these genes, both by activating beta cell genes and repressing alpha cell genes. So how is it working in the alpha cell then? So what we were able to do, we got this beautiful glucagon Cree line from Shota, and who's I think also in the audience here, which is improved Cree. So it's a glucagon, it's not inducible, it's improved. And we knocked out NKX2.2 just in the alpha cell. Obviously, I think everyone knows that it's really hard to study a lot of molecular studies in the mouse alpha cells because there's so few of them, so we also used an alpha TC culture cell line to do some of these studies. And I'll try and point out what the data is from. So just to show you that this Cree, so it's been a long time, and this is what I think held back some of the studies in alpha cell in mice, is that we didn't have a good specific Cree. So I just wanted to take some time to show. This is, we had generated NKX2.2 floxed mice with the glucagon Cree, and this is just a tomato reporter, and you can see the tomato reporter is really nicely specific in the glucagon expressing alpha cells, and it's not expressed in the beta cells. So it's a really nice Cree, and we could actually show that we were able to knock out NKX2.2 in the cells that actually were the alpha cells and where Cree was expressed. So obviously we had a phenotype, otherwise I wouldn't be showing this to you. So we did see in these cells, so we're just showing here the flox control and the Cree control, so we have normal number of alpha cells, and in the NKX2.2 knockout we had a reduced number of alpha cells. Unlike the null, we do still see some alpha cells formed, and we can talk about that. But we had about half the number of alpha cells, whether we just looked at total alpha cell area or the number of cells that were actually expressing the CRE. Again, we didn't lose all alpha cells. Let's see. But what we saw was in these, where we lost the alpha cells and glucagon expression, we actually started to see beta cell genes coming on. So very similar to what Marcella was showing in the type 1 diabetic islets. So here is actually a good example here. Here is a glucagon and insulin co-expressing alpha cell. And we did the lineage trace as well. So we know they're alpha cells. And you can see we had a significant increase in the number of cells that now co-express both glucagon and insulin. In addition, we saw an upregulation of somatostatin. So again, these alpha cells are losing their identity. They're turning off glucagon, or they're even keeping glucagon on, but now they're expressing the other non-alpha cell hormones. So this is, oh, and then we also looked to see it did have a phenotype. In mice, we don't have a very robust phenotype when we lose alpha cells, but we did see fasting blood glucose levels were actually reduced, and the mice were very insulin sensitive. So how is this working? As I said, in beta cells, the same transcription factor is activating beta cell genes and repressing alpha cell genes. But now we're seeing the exact opposite happening in the alpha cell. So how is it actually functioning in opposite ways in the same cell, in the different cell types? So what we did is what most everyone does, is we basically sorted out the tomato positive cells. So these are the glucagon or the alpha cell lineage cells, and did RNA-seq. And what we did is we compared that to this data set that Mark Housing had generated, where he sorts each of the islet cell populations. So we can basically compare to delta cell genes, beta cell genes, and glucagon genes. And when we did this comparison, we looked to see, okay, what was NKX 2.2 regulating within the alpha cell? So we've sorted out the alpha cells, and now we're comparing it to all cell populations. What was really cool is that what we saw was, so here's just alpha cell genes, and there were very few that were upregulated when we knocked out NKX 2.2, but not surprisingly, a lot were downregulated. So again, it shows that NKX 2.2 is activating alpha cell genes. But what's really interesting, again, is that we saw this huge increase in the beta cell genes and the delta cell genes. This is, like I said, completely opposite to what we saw when we knocked out NKX 2.2 in the beta cells. So how is this working? This is just showing a number of the genes, and I want to point out several. MAFB, IRX2 is another alpha cell gene, glucagon itself, and then here we have MAFA, insulin. So we're basically, again, converting these cell types. So for us, this was really interesting, but it's still a puzzle. How is it doing this opposite regulation in these two cell types? And I did want to point out, actually, I added this in because this is, what we did is we took the genes that were changed in the NKX 2.2 alpha cell knockout and compared it to Marcella's data set from T1D alpha cells. And what we see is about a 10% overlap. So NKX 2.2 is actually responsible for a lot of the gene changes that are seen in alpha cells in type 1 diabetics. And again, this is just showing a number of these different genes, including MAFB. And I wanted to point out MAFB. We actually verified this. So this is Marcella's data where she sees a decrease in MAFB. And we see a decrease in MAFB not only in general, but also there's a lot of glucagon expressing cells that are still there that are no longer expressing MAFB. So we've basically downregulated many of the genes that are seen that are downregulated in type 1 diabetes. So how is this doing this? And again, like I said, this is just summarizing. We see in alpha cells, NKX 2.2 is responsible to activate alpha cell genes and repress beta cell genes, where the opposite is happening in beta cells. And we just, to try and understand, well, actually, we tried a number of experiments to figure out how this was working. In the end, we did what I would say was one of our few aha experiments, is we just did a chip-seq, and it turns out that, remember I told you that in beta cells, NKX2.2 predominantly binds enhancer regions, but in alpha cells, it actually targets promoter regions. So you can see here, these are all promoter regions in the alpha cell, and you can see there's a high enrichment in alpha cells of NKX2.2 binding promoters rather than enhancers. And this is beta cells, we hardly see any promoter binding. And this is just showing it the numbers here. So again, the number of peaks at the promoter regions was very much increased compared to the beta cells. So how is it targeting different regions of the genome in these different cell types? So what my student did is he mapped where all the NKX2.2 binding sites were, and then he looked nearby in enhancers to see what other transcription factors could be binding nearby. And this was supposed to be animated, but we can do this. So, and then he said, okay, let's just look for those transcription factor binding sites that are either in just beta cells or in just alpha cells. And lo and behold, there were 5 actually transcription factor motifs that came out of this, but the one that really excited us was KLF4. So KLF4 was actually not described as being in alpha cells before, but is very alpha cell specific. And you can see it's enriched a lot in the promoters that actually have NKX2.2 binding of all of the different transcription factors. ETV1 is here. It's also an alpha cell specific transcription factor, but this was much more significant. And we got excited about KLF4 because, as probably most of you know, it's a really well-characterized Yamanaka factor. So it's basically involved in being a pioneer factor and opening chromatin. And it turns out it's very alpha cell enriched. So we can detect a little bit in beta cells, but it is highly enriched in alpha cells. And so what we found was when we looked at all of the attack-seek peaks, so we knocked down KLF4 in an alpha cell line, in the alpha TC cell lines. And when we did that, we looked at the differential accessibility peaks, and it turned out that we lost accessibility where NKX2.2 was binding. And then we also knocked down NKX2.2 in the alpha cells and knocked down KLF4, and we saw a number of genes that had overlapping differential expression, including MAFB and IRX2, these 2 really specific alpha cell genes. So this is just showing the chip alpha in alpha TCs of NKX2.2, and here is the NKX2.2 binding this promoter region of MAFB in alpha cells. You can see it actually doesn't bind at all in beta cells. And this is the alpha cell attack-seek. And when we did the chip for NKX2.2 is just showing here, and when we did the chip for KLF4, it's overlapping. So this is actually, you can probably barely see it. This was a documented NKX2.2 site, but now we know that KLF4 also binds there as well. So what we're thinking is happening at the MAFB promoter is that KLF4 and NKX2.2 are co-binding to activate the expression of MAFB. And in beta cells, you don't have KLF4 or NKX2.2 binding. You have closed chromatin, and MAFB is not expressed. And this is just showing, again, IRX2, the same thing. So it wasn't just the one gene. Here we've got NKX2.2 binding the promoter, no promoter binding in beta cells, and then we have open chromatin in the alpha cells. And NKX2.2 and KLF4 are also co-binding at this promoter. So this is saying the same thing. So what our hypothesis is that KLF4 as a alpha cell specific pioneer factor is opening chromatin at promoters of alpha cell genes and then allowing NKX2.2 to come in and bind and activate those genes. So the question is, what if we can misexpress KLF4 in beta cells and see, do we actually activate alpha cell genes by allowing NKX2.2 to now bind? And so we did this expression, again, using a cell line. So we used the Min6 beta cells, and we either used an empty vector control or we overexpressed KLF4 in the beta cells. And it was really exciting. This is an experiment, actually, the thesis committee told us not to perform, but when you look at this, we see an upregulation of all these alpha cell genes in beta cells now. including ARCS, MAFB, IRX2, and this is just showing that, yes, KLF4 is now misexpressed in these cells. And this is just showing, again, it's very highly significant. We see we upregulate KLF4, and now we have expression in beta cells of MAFB and IRX2. Interestingly, we did not see a reduction in the beta cell genes. Now, this could be because it's a cell line, but it could also be that KLF4 is only responsible for opening chromatin to allow NKX2.2 to bind in a different genomic region. This is just showing, again, just MAFB locus. So you can see normally we don't have open chromatin at MAFB in the beta cells, but if we overexpress KLF4, it becomes open, and now we activate the MAFB gene. And we see an increase in NKX2.2 binding at this promoter. So we do think that overexpression, or KLF4, actually facilitates the expression of NKX2.2 dependent alpha cell genes. We can also deplete, so that's KLF4 is actually sufficient to actually activate alpha cells, but we can also use alpha cell TCs to deplete KLF4, and we again see a downregulation of alpha cell genes. We see some upregulation of beta cell genes, but not as much. But when we did this in a mouse now, we again knocked out, using the glucagon Cree, we knocked out KLF4, and again, now we see both a decrease in alpha cell genes and an increase in beta cell genes. So this was really exciting, and I'll just summarize here, and then I'll talk about where we're going to this with the human. So we know that NKX2.2, in addition to regulating beta cell functions, is very important for alpha cell functions. And we think it's doing this by binding promoters rather than enhancers in the alpha cells, and this is facilitated by KLF4. So KLF4 and NKX2.2 seem to cooperate to regulate the alpha cell program. And in beta cells, since we don't have KLF4, NKX2.2 is not binding at these genes. And we've been able to show that KLF4 actually facilitates the activation of alpha cell genes in beta cells when we misexpress it. So what about human islet development? So I've told you about our studies in mice, but we'd love to study this in humans. Obviously, it's more difficult, but we have set up a system where we have knocked out NKX2.2 in an ES cell line and then seen how it affects differentiation. And this is just, we're using the standard multi-step protocol that everyone's using, and just to ask, okay, what happens when we lose NKX2.2 in actually, we're knocking it out at the very beginning at this point. And was really nice to see, although maybe not so much of a surprise, but we do see a loss of insulin-positive cells, glucagon-positive cells. And actually what was a surprise to us, we also see a loss of somatostatin, but the cells that still express insulin also express the other hormones. So they're really becoming polyhormonal, just like we were seeing in the mouse. Interestingly, KLF4 is not in the human alpha cells, but KLF5 is, and we see a downregulation of KLF5. So we're thinking maybe KLF5 is actually serving to function in the same manner as KLF4. And when we looked at Marcella's data, again, both KLF5 and KLF8 are downregulated in her data set in the islets from type 1 diabetics. And this paper that just came out showed that KLF5 was also downregulated in patients with type 1 diabetes. And so we're hoping that we've now identified really maybe one of the master regulator proteins of this alpha cell identity that is getting lost in type 1 diabetic. And so again, this is where we're going. We're going to start looking at the KLF proteins in humans, but we really are excited that we're starting to, I think, understanding not just how the alpha cell identity is made in the first place, but how it's getting lost in conditions of diabetes. And we really think these transcriptional regulators are key in regulating that decision. So I think I actually finished on time. And I just want to point out, Elliot Brooks, this was his thesis project. He's defending next week. And he was helped by a really talented undergraduate student. And now, actually, we've got Chris is starting to do the human ESL work. And obviously, I need to thank my funders and you for your attention. Beautiful Denver on a morning run.
Thank you, Lori, for a very wonderful talk. Our second speaker for this session is Joan Camuñas, did I say it correctly? Soler. He's an assistant professor of bioinformatics for translational medicine at University of Gothenburg.
Thank you very much to the organizers and to the Helpline Foundation for inviting me to give this talk, as well as to the chairs of these and next sessions. And I'll focus my talk on transcriptomic studies. I'll first discuss briefly what have we reached in the field, and I'll then go down some of the studies that we've done during my postdoc and now in my lab, where we focus in the function of alpha cells and what can we learn by coupling transcriptomics to other methods. So first of all, in the last decade almost, single-cell techniques, and especially single-cell transcriptomics, has allowed us to obtain unprecedented molecular characterizations of cells in the human body, and mostly this has been used in most fields to refine cell type identification, so find new cell types in tissues, and also discover heterogeneity in specific cell types by coming up with subtypes or variation inside cell types that we thought that were homogeneous, and identify all of this molecular heterogeneity that was not seen before. And of course, this has been widely used in the field of pancreatic research and in the study of islets. Here I show you an image of islets where maybe you might notice that there are a lot of alpha cells, and I will not discuss this in detail, but I think that a few groups are finding now that there's variety within islets and the amount of cell types that we see. But essentially, the use of single-cell transcriptomics to study the islet has evolved really, really fast. So the first studies came up in around 2016. And these were studies with a few thousand cells and less than 10 donors, most of them. It quickly evolved into not only doing single-cell transcriptomics, but also looking at gene regulation with the tag-seq. And very quickly, it moved into multiomics, where you get the two layers at the same time, transcriptomics and regulation. And right now, we're in the stage where these studies are moving from less than 10 samples to up to hundreds of samples, mostly thanks to community efforts like the HPAP, and where we can start to tease heterogeneity across multiple types of donors and patients, and get really deep down in the characterization of the islets transcriptomically. And this is, I wanted to show you, start with an image from a paper from HPAP, summarized in a study by Kyle Golton's team. And I think it reflects what we can achieve with large samples, which is try to understand this heterogeneity in the islet cell types and the cells that surround them. And one thing that you might notice here is that when we try to identify genes that are differentially expressed between disease conditions and non-diabetic donors, there's a lot of variation on what studies find. This is, I think, the picture of the largest data set to date. There's about over 60 donors and more than 200,000 cells. And apparently, according to these results, most of the variation or most of the differential expressed genes in type 2D would be found in beta cells, a few in alpha cells, but not so many. This is surprising to me, and I think that this might change with time, and a lot of variation in many cell types when we look at type 1 diabetes and autoantibodies. And this brings me to the next slide, is that single-cell transcriptomics is great, but actually most studies that have tried to put together what we find in different labs and different groups and different papers find very little overlap. So this is from a review a couple of years ago, where the overlap in differential expressed genes for beta cells and alpha cells between different studies is really, really small. And I think this is an issue for all of us, at least that work in this field. And for a while, we've been discussing that some of the issues with transcriptomics is potentially sensitivity of single-cell sequencing. I think that this is less and less the case, because methods are getting better and better. And of course, we cannot capture every transcript in a cell, but we can get very good representations by studying at the same time many cells of a same donor. And I think that most of the big things will come through the integration of large sample sizes. I think that most of the studies so far had still relatively small sample sizes if compared to fields that have matured earlier, like GWAS studies. And of course, it will also come through the use of more and new integration methods that allow us to model the donor variance and heterogeneity. And these will benefit a lot of all of these large data integration projects that are happening now and that are making these large data sets available to the community. And I think another issue that has already been brought before by all powers is the heterogeneity that happens both in the disease, so not all patients are the same. Every patient are different, and there's heterogeneity in the patients, and also potentially heterogeneity within the cells, and this is not always easy to tease when doing differential expression analysis. And finally, there are some factors that are difficult to avoid, which is the dissociation into single cells, and these will likely change fast as new methods to work in tissue slices and potentially by working with full organs are evolving to study molecularly these tissues. And I wanted now to move into the thing that I've been interested in the last few years from my postdoc and now in my lab, which is using physiology and cellular function to couple it to transcriptomics and try to use these two layers of information to access some more information that can be usually obtained in standard single-cell methods. And the idea behind this is that by doing so, we might be able to obtain some phenotype of the cells that we can place on top of the transcriptomic information, and this can be used to look at covariation between the two types of data sets, and hopefully the idea is that this can help us tease apart some of this cellular heterogeneity and see if it correlates to functional variation and to dysfunction in diabetes. And I think where this is heading and what I'm excited to work in my lab in the next years is try to bring some of these things when we look not only at dissociated cells, but also when we put it together in a tissue. And this is what can we look at when we try to look at function in islet cells? This is an image actually from a beta cell, but surprisingly enough, most of the pathways that regulate insulin and glucagon secretion are shared. There's a whole session on Thursday discussing glucagon secretion itself and how it differs from the secretion of insulin by beta cells. But one such thing that it's essential for the secretion is the electrophysiology of these cells, so the electrical responses that start triggering the release of vesicles. Another proxy of function of these cells is calcium imaging, where for alpha cells, we would see calcium responses at low glucose, whereas for beta cells, these responses happen at higher glucose conditions. Another thing that one could track is vesicle or hormone content, and finally, secretion of hormones, these cells here characterized with insulin trace. And the work that I developed for a few years focused in the electrophysiology and combining with transcriptomics. This was done with the team of Patrick McDonald in Canada. And we did all of these experiments in isolated cells, pancreatic islet cells, alpha and beta cells, that we could measure electrophysiology and do patch clamp sequencing and then single-cell transcriptomics. And couple both types of information, the transcriptomic information and the electrophysiological information at the same time in the same cells. And one thing that I will come up later is that in most of these studies, it has been always easier to start analyzing the beta cells. And the reason for this is that there's a lot of, we observe much more variation in alpha cells in general, and it's usually harder to understand what is happening. But in this first study, we use it mostly to characterize beta cells and find genes that would correlate to the exocytotic parameters of cells at different compartments of the cell, identify genes that we could validate with knockdown experiments, and finally identify what we thought was a maladaptation of beta cells in type 2 diabetes, where we saw that whereas beta cells were upregulating genes that seemed to correlate with exocytosis, they would failing to do so, and they actually responded worse. After doing this, we looked into the alpha cells, and by looking at electrophysiology of the alpha cells, we found actually quite surprising, and this is based on data of 30 donors, but Patrick's team has data on many more donors where they observe the same type of responses in isolated cells. And this surprising result is that actually in cells from non-diabetic donors, we observe what you would expect, which is higher exocytosis at low glucose that decreases as you increase the glucose conditions. However, for alpha cells from donors with type 2 diabetes, we observe the opposite. So at high glucose, these cells would respond higher, and at low glucose, they would not respond enough. And this, we think, is something that could contribute to the impaired response of the alpha cells, and of course, all of the problems associated with hypoglycemia. So the question here is, like, why is this happening, and can we use any of these techniques to learn which pathways might be going wrong, specifically in these cells that are not responding here? And to do that, we use the same repertoire that worked for beta cells and where we had an easier ground to validate our findings because we know more about beta cells than alpha cells, whereas in alpha cells, we know less, and therefore it's sometimes difficult to know if what your finding is to validate it and see if it agrees with other techniques. And what we noticed quite quickly is that when we look at correlations of genes with total exocytosis in non-diabetic, we found more associations at low glucose. So when we look at low glucose, we find more genes that are correlated to higher secretion, which seems reasonable. And then there's a shift towards anti-correlation at higher glucose. To try to identify factors that could be important for this response, we look at the edges, at the tails of these distributions. And surprisingly, what we notice is that the genes that pop up as correlated to exocytosis at low glucose are the same that come up anti-correlated to exocytosis at high glucose. And these ones were mostly found in the mitochondrial respiratory chain complex and in other factors that seem to be involved with the mitochondria itself. So we identified other pathways, doing a pathway analysis, but mostly we came to the conclusion that mitochondrial respiration might be important to set the responsiveness of alpha cells in their normal or physiological behavior. When we next looked at alpha cells from donors with type 2 diabetes, we found that these two distributions were flipped. So actually now we found more genes correlated to secretion or wrong secretion at high glucose, whereas the distribution at low glucose is shifted towards anti-correlations. We were interested at looking at this region here. We could not find genes that tell us why some cells are not secreting enough low glucose in this type of analysis. But we found that actually over exocytosis in high glucose was mostly associated with immaturity and markers of early development in alpha cells. So that gave us a lead to keep looking into these genes and into these pathways through other analyses. Actually, to do that, we flipped to another approach. One way to analyze this data is through correlations, obviously, but then there are other techniques to try to extract information from these complex data sets. And here we work together with Linford Bryan in Oxford, and he's an expert in developing models to classify cell types not from gene expression, but from electrophysiology only. And he helped us develop models that could predict the cell type based on the electrophysiology, which for human cells is not straightforward. The first models we developed were mostly using the cell size, which alpha cells are smaller than beta cells. But then we removed this parameter, and we could fit and predict cell type with quite good accuracy, here around 90% good classification, using exocytosis and other types of currents of the cells. And the surprising thing here is that when we try to transfer this model from the learning set that was mostly non-diabetic donors to the type 2 diabetes donors, we found that the model was performing quite well in most cells, but it would fail in a few cells where the model was thinking that they were completely the opposite cell type. And we did 3 different variations of this model that didn't use cell size. And the 3 of them gave similar results. So the model predicted well, mostly in non-diabetics, also in test sets. But in type 2 diabetes, it would fail and think that some alpha cells behave more like beta cells. And the model was quite certain of putting them in the wrong category. So the first analysis we did here is like select these cells here and run correlation analysis with the probability that the model calls them an alpha or a beta cell. And again, we found that the cells where the model was performing worse were enriched in some immaturity or some transcription factors that are mostly connected to lineage. So we started seeing genes like NeuroD1, IL-1, FIP, ARCs, and genes that are involved with lineage in alpha cells. To learn more about these, we then focused in doing transcriptomic maps, UMaps on these genes. We found that we were noticing quite a bit of heterogeneity in the expression of these genes in our single cell data set. This is something that needs to be seen in larger data sets, but at least with patch-seq data, we could find quite a bit of heterogeneity. We found that it looked like there were two different subpopulations, especially in the type 2 diabetes group, with differences in expression of these genes. And specifically, when we would run the model in cells that had either high or low expression of these key transcription factors like ARCs, we found that the model actually failed only in cells from type 2 diabetes donors, which had high expression of these transcription factors. So the model was performing well in cells that didn't have high expression of lineage factors. And actually, if we focused not only in the modeling results themselves, but on exocytosis, these cells that have high expression of lineage factors were the ones that were not responding well at low glucose conditions for type 2 diabetes donors, suggesting that some higher expression of these factors might not be beneficial for alpha cells, actually, and might be connected to a lower functionality in these cells. And we then validated some of these findings by doing immunostaining, where we could see this type 4 groups of subpopulations of cells with expression of the different transcription factors in themselves. So overall, with this study, what we learn is that we think that patch-seq and coupling of functional measurements with transcriptomics can help us learn transcriptomics out of the functional readouts by doing this coupling. We found links to maturation, respiration, and expression of certain receptors with alpha cell function. And we found that alpha cells that are not functioning as we would expect them to do in type 2 diabetes, they were associated with a beta cell functional phenotype, and that decreased exocytosis was happening mostly for cells that were enriched in immaturity markers and lineage factors. So that's what we could learn by using patch-seq in these alpha cells. And now in work I'm doing in my lab in collaboration with other teams in GU and Oxford, mostly Patrick Rosman and Anders Rosengren, we are trying to develop other techniques which we could use to do similar type of measurements, but where we can do this at a higher throughput. And the approach here that we thought is based on an earlier work from Patrick's lab. And the idea is that whenever an endocrine cell secretes and releases a granule of insulin or glucagon, by connecting this vesicle to the membrane, it exposes actually to the extracellular milieu. So we added a dye. We can add a dye, a dextran, a small sugar, and then this dye will just diffuse inside these vesicles. And after secretion, these cells need to recover all of this membrane that they have added to the cell surface, and therefore this dye will end up being internalized, and these cells will become fluorescent. And potentially, we think that we would be able to track exocytosis by measuring this recovery of endocytosis later on. And by doing experiments with full islets, we find that this seems to work. We can see increased fluorescence in beta cells when we submit them to stimulatory conditions protocols. We don't see that at low glucose. And we have, I don't show it here, but we're generating new data showing that something similar might be actually happening for alpha cells. So potentially we could also track glucagon secretion. And this is useful because then one can pass these cells through FACS sorting. This fluorescence is stable over a longer time period. And when we do this on cells that have been submitted to this stimulatory protocol, this is again for beta cells, we see a shift in the fluorescence of this tracer, and we see an enrichment of cells that have high fluorescence and that therefore have been internalizing the dye, presumably because they've been secreting insulin. Then the idea here is that we can fact sort these cells, and we can do apply the same repertoire of tools that we developed for PATCH-seq and try to identify targets that correlate with this compensatory endocytosis protocol. And this is work by Vishal in Anders' team and Marta postdoc in my team. And again, it's much easier to start with beta cells because we know so much more, and results are in generally easier. And what we found, we started working with mouse models. We had wild type, DBDB as an extreme model of diabetes, and then fumarase KO, which has impaired mitochondrial responses, which is developed also by Patrick's team. And essentially, this allows us to characterize the different cell types of the islet. We find that beta cells from wild type and fumarase are more similar, whereas DBDB is really far away in gene expression. We find that the fluorescence also of this tracer behaves as we would potentially expect. There's a good response to glucose for wild types, whereas this is impaired in reducing fumarase and this completely impaired in DBDB. And then we can start doing all of this characterization again on these cells. We can see that cells from the 3 mouse model are transcriptionally different. We can characterize them with different gene pathways. And what was surprising to us is that the genes that characterize the wild type beta cell actually decrease in expression in the fumarase model, which is a mild model of diabetes, and they are really low in expression in the DBDB model, suggesting that this is going downhill in the disease. Again, we can apply a correlation analysis, and here what was more remarkable is that for the wild type, we found mostly genes connected to higher exocytosis, which it seems to tell us something, because in general, in the DBDB, we find more genes anti-correlated to exocytosis, whereas in the fumarase model, we find an equivalent number of both types of genes. So somehow it's easier to find genes that correlate to function in cells that are functioning correctly. And here is an example of one of such correlations where we see no correlation in the wild type. We see negative anticorrelation in fumarase and a stronger anticorrelation in DBDB. And again, when we look at the genes that pop up here as associated with good functionality in ND, we see mostly beta cell genes that are connected to insulin signaling. For the good response in the fumarase model, again, we see mostly representative genes that are lineage factors of beta cells. We realize also that mitochondria comes up again here, and this is something that you cannot capture in pathway analysis, but we can, by looking at the genes themselves, we can look at and find them. And when we start looking at this function in these 2 disease models, what we find is that there is an enrichment of glycolysis and gluconeogenesis, as well as pathways that might be involved in protein generation, like the ribosome. Whereas in the DBD model, we see much more pathways related to antigen presentation. We then connected all of this information into a plot where we focus in pathways that are important for beta cells. And what we notice is that mostly there's 3 pathways that seem to show dysregulation, which is the UPR and ROS defense, where we see that as we go through the 3 disease models, we lose correlation between the gene expression of these genes and exocytosis. And we can validate this in islets from 15 human donors, where we found the same trends for these 2 exact pathways. We're now doing functional validation of some of these genes. And just to show you more on the human data that we collected for beta cells, we can see in humans that there's also the expected response, an increased response at high glucose, similar to the mouse models. And these are the pathways that we see to change with human diabetes. And we are now trying to integrate this into a bigger pathway analysis where we can connect and look at pathways that show, for instance, dysfunction in type 2D, and that they are concordant between the mouse model and the human data, or pathways where we see human-specific effects, so we see concordance only in humans, but the mouse models do not correlate. Again, here you might see that we have a lot of pathways popping up with the strongest model of disease, which is the DBDB, and this might be related to the age of the mice themselves. What might be more exciting for this meeting is that we are now trying to extend this to alpha cells as well. It is harder. We don't have that many cells, and we are growing the data set, but just to show you things that we are finding, this is from the last few weeks. The responses are not as clear in human cells. For mouse cells, it works better. For human cells, what we notice is that in type 2D, the compensatory endocytosis seems to be much bigger all over the range of values that we can measure. So there seems to be a much broader range of compensatory endocytosis in these cells from donors with type 2 diabetes. We need to collect more data on this to see if this holds. And when we look at correlation, so far we've identified some genes that show significance mostly in hormone processing. And one of the genes that show the highest significance was a negative regulator of PCSK1, which it's mostly involved in the processing of insulin, but we are wondering if it could play a role in the alpha cell or could be giving it a more of a beta cell kind of phenotype. And just other work that is being done in my lab nowadays is working also trying to bring some of these measurements in tissue slices. So these are slices from a non-diabetic donor where we look at calcium imaging of this tissue, and we are trying to identify beta and alpha cells by looking at the responses at different stimuli like glucose or amino acids. And we can collect traces, analyze the number of events that we see in these different protocols that we apply. This is samples that we obtain from Uppsala that can provide live human tissue on really quick after 1 day of isolation. And so far we were lucky enough to receive one sample of one donor with type 1 diabetes. We were also able to measure some of responses on some cells in this tissue, and we are trying to couple this to immunostaining. Here you can see an islet where there are mostly alpha cells, but also one beta cell. And Sandra, which is my postdoc, will give a talk in the DPG meeting on Vienna on this topic. And that's the summary slide for this part of the work. We are trying to develop new tools to do tracking of the secretory history of islet cells by compensatory endocytosis. We think that we can use this method to do higher throughput studies on genes that connect to secretion in these cells. For beta cells, we found links to mitochondrial respiration mostly in dysfunction in type 2D. And we are now validating this methodology to also study the alpha cell, where we mostly see links to hormone processing. And ideally, in the long run, we want to bring some of these results in tissue by studying live pancreatic slices. And thank you all to all of the attention and all of the collaborators of this work, as well as the funders and organisms that have provided us with tissue. Thank you.
All right, thank you very much.
Thank you.
And thank you all for coming to this first session. I believe we have a coffee and snack break.